Hello students, welcome back. Today we are going to learn the basic concepts about genes. So first of all, we'll see some definitions of gene. Gene is nothing, it is a specific sequence, a specific part, small portion of DNA which codes for protein, that is, it contains genetic information. So you can say it is a specific DNA sequence that contains genetic information. The gene is unit of genetic information that specifies synthesis of polypeptide. That is, it has messages in it which is further translated into protein. The unit of genetic information that controls specific aspects of phenotype. That is, it is a unit of heredity. It controls, as it has messages in it, it finally controls the phenotype, and thus, as the genes are transferred from parents to offspring, it is a unit of heredity. History. The term gene was coined by Johnson in 1909. E.R. Garrett in 1908 proposed one gene, one product hypothesis. That is, one gene will produce one product. Gene theory was proposed by T.N.Rao College in 1911. In its theory, he proposed that chromosomes contain hereditary units and each chromosome carries hundreds to thousands of genes. So inside chromosome, DNA, chromosome is made up of nucleic acid and protein, and genes are present in it. So each chromosome, single chromosome contains many genes. That is, number of genes far exceeds the number of chromosomes. He again added that the genes are arranged on chromosome in a linear order and at a specific site, and that specific site is called locus. Pauling and Ingram in 1949 established the role of genes in protein synthesis. Now we know the process, replication, what is replication, transcription, and translation. But at that time, Pauling and Ingram established the role of gene. It transcribed to produce mRNA, and translate to produce protein. Weidel and Tatum in 1948 proposed one gene, one enzyme. That is, one gene will produce one enzyme. That was proposed by Weidel and Tatum. And the fine structure of gene was given by Benzer in 1960. Structure of gene. Mainly, gene contains three basic components. One is coding region, which is called exon. Exon, it specifies the sequence of amino acids, while introns are non-coding sequences which do not specify amino acids. Third is regulatory sequence. It plays regulatory role. You can see over here in diagram. These are exons. These are exons, and in between two exons, introns are present that are non-coding region, which are omitted or which are spliced out during post-transcription process. Upstream to the gene. This is a complete gene, whole complete gene. Upstream, this portion is called upstream, and this is called downstream. The right side is called downstream, and this is called upstream. To the upstream, these are the regulatory sequences which play or which regulate the processes. Now, certain important features of gene. First of all, location of gene. So we know in a eukaryotic cell, within nucleus, chromosomes are present, and within chromosome is made up of DNA and protein. Within DNA, genes are linearly arranged in it. Genes are transmitted from parents to offspring, and that is why we call it as hereditary material. They are responsible for character of organism. And the site at which the gene is present in DNA, that specific site is called locus. Now, we have discussed this earlier. Many genes are located on a single chromosome, That is, number of genes far exceeds the number of chromosomes. Genes are arranged in a single linear manner. Genes can undergo mutation, that is, a sudden heritable change is called mutation. Gene can undergo mutation. Gene can even undergo recombination process. If the position of the gene changes or the sequence of the gene changes, finally the effect is seen in phenotype, that is, the character also changes. Genes may exist in several alternative forms called alleles. We already have discussed this. What are alleles? Allele is an alternative form of same gene in a diploid organism at, at specific site, at specific locus. A specific gene is present in one chromosome. Same gene will be present in same another chromosome at that particular site. If that gene shows same character, but its effect is different, then such at such locus or such genes are called alleles. Gene can undergo duplication process, that is, it produces its own digital copy. Process is called replication. Genes are also capable of recombination. A gene is a particular DNA segment which contains the information to synthesize polypeptide chain, that is protein. This process is called translation. As you can see in diagram, this is the nucleus. Three main important processes of central dogma of life is replication, transcription, and translation. Replication and transcription in eukaryotes occur in nucleus, while translation which occurs in cytoplasm. So here you can see this is a gene. Transcription, mRNA is produced by transcription process. After that, through post-transcription modification process, RNA introns are spliced out, that is, unwanted or you can say non-coding region is removed by RNA splicing process, and this a complete mRNA is then exported from nucleus to cytoplasm, and it reaches at the site of protein synthesis, that is at ribosome. And here protein is synthesized. So a gene, within gene there is a messages, and that message is codes for particular protein. Now this information which is present in gene is contained as a sequence of nucleotide which is called genetic code. The information is nothing, it is a sequence of nucleotide which we call as genetic code. And now that sequences are of 3 nucleotides, that is a triplet code for a single amino acid, and that triplet is called codon. So there are many codons which codes for particular amino acid. It is given in this table. These are different codons which codes for particular amino acid. You can see over here UAA, UAG, that are stop codon. There are certain start codons. This AUG codes for methionine. So these are different amino acids which are coded by a particular codon. Now, another definition is gene is regarded as fundamental and indivisible unit of genetic information in terms of structure and function. That is, gene is the smallest unit. It cannot be further divided. That is why indivisible unit of genetic information. So, further, it was challenged by recombination studies in bacteriophage by Benzer, and he showed that it could be subdivided. Gene could be subdivided into smaller units, that is muton, cistron, and recon. Muton is the smallest part where mutation can occur. Recon is the smallest portion where recombination can occur, and cistron is the smallest, or you can say it is a smallest part of function. Function means which codes for protein. So important terminology, cistron, it is called unit of functions. That portion of gene which can code for protein. The region which do not code for protein is not included in cistron. That is, introns are not included. So, cistron which can synthesize polypeptide, which can code for protein, the smallest unit that transmits genetic information. It is an alternative term given to gene in case of protein. So you can say cistron is the smallest unit which transmits genetic information because it contains codons within it. It possesses messages in it, and that is why it can be transferred from generation to generation. But in case of prokaryote, as introns are not present, a gene is not interrupted by non-coding sequences. You can say cistron is an alternative term of gene in case of prokaryotes. So in prokaryotes and in lower eukaryotes, there is usually a simple relationship between gene and its product. Relation is equivalence. Thus, gene and cistron are equivalent. Gene is a unit of function, or you can say cistron is a unit of genetic function. Both is same in case of protein. You can see in diagram here. This is gene 1, gene 2, gene 3. And in between each gene, internal UTRs are present. That is untranslated region. But within gene, non-coding sequences are not present. This complete gene, it codes for one complete protein. That is, this protein is directly proportional to the sequence of gene present. And that is why this gene itself is called cistron in case of protein. But while in case of higher eukaryote, gene contains introns. That is, a complete gene is interrupted by non-coding sequences, which are removed during post-transcription modification. That is, they are spliced out, and finally, final product, that is protein, do not contain the sequence of introns. That is, cistron and gene are not similar in case of eukaryote. mRNA, that is the final mRNA which codes for protein, is actually the copy. Protein, the sequence of protein is exactly similar to the mRNA sequence, and that is why that portion which directly codes for protein is called cistron in case of eukaryote. So, which most eukaryotic genes contain introns, which are then presented, which are then not presented in final. Therefore, it is not part of its function. Introns are not part of the function, that is, they are not included in the protein. Thus, the cistron is therefore equivalent to exon of eukaryotic. So in eukaryotes, cistron is equivalent to exons, while in prokaryotes, cistron is equal to gene. So you can see in diagram, these are exons, that is coding region, and these are introns, that are non-coding region. And this complete portion is called gene. So in gene, coding sequences are interrupted by non-coding sequences. Transcription occurs. After transcription, this RNA is produced, and post-transcription modification occurs. And non-coding sequences, they are spliced out. Here you can see in this mature mRNA, non-coding sequences are not present, and this mature mRNA is then converted into. So exons actually combine to form a mature transcript, and that mature transcript produces protein. So exons are proportional or equivalent to cistron in case of eukaryotes. Thus, you can say that prokaryotic gene is uninterrupted. They are not interrupted by introns. In eukaryotic gene, the coding sequence, that is exons, are separated by non-coding sequences. There are introns. That is, eukaryotic gene is interrupted by introns. In complex eukaryotes, introns account for more than 10 times as much as DNA as exons. Thus, in eukaryotes, what it is observed that introns amount is higher. That is, it accounts for more than 10 times as much as exons. So, this was the overview of gene. We are ending our lecture here. We'll meet soon. Till then, stay home, stay safe.